I'm delighted to welcome Professor Wendy Barclay of Imperial College London to deliver our first prize lecture of this conference. The prize medal is awarded to an outstanding microbiologist who is a global leader in their field and whose work has had far-reaching impact beyond the discipline of microbiology. I can safely say in this case we have an outstanding recipient in Wendy, whose research has focused on respiratory viruses and the factors affecting how they are transmitted and cause disease. She has contributed to the understanding of how these viruses cause pandemics and how we can best develop strategies to combat them. It's my honor to introduce Wendy, whose prize lecture is titled Perspectives on Pandemics.
Well, thank you very much for the kind introduction and a huge thank you for this award. I am completely humbled to receive this award. There are many people sitting in the audience today who are equally, if not more worthy than me. I feel like I'm happy to accept it because it's very lovely to see that a UK microbiologist has been given this award this year. And I do hope that sets a trend for the future. And I also feel that UK virology and the community that we have has really pulled together in the last 3.5 years in the face of a pandemic. And I guess for those reasons, I'm very happy to accept the award on behalf of all of us. So I'm going to be speaking about pandemics. But before I get to that, of course, I need to thank various people. These 5 individuals I consider to be principal mentors and collaborators that have really shaped my career, all the way from my PhD supervisor, Dr. David Tyrrell, who sadly isn't with us anymore, through Jeff Armand and Peter Palese, my postdoctoral mentors, and then to Maria Zambon, who's been a lifelong now friend and colleague, and Ray Pickles, who have influenced the way I've thought about what we do. But really, the award also goes very much to the people I work with in my group and have done for the last 27 years since I set up as an independent PI. And I did have a great big list of names, and in the end I decided just to show photos because I think they really encapsulate what I hope doing science in the Barclay group is all about, which is being friends, working hard together, but having lots of fun together as well. So it would be astounding, I think, in 2023 if you went anywhere in the world and the average person on the street didn't know now that animal viruses can infect humans and cause us problems. When an animal virus infects a human and doesn't really go much further, we call that a zoonosis. But sometimes those viruses mutate further and adapt to the new human host, and then they cause pandemics. And of course, we've seen that so vividly in the last 3.5 years. The sorts of questions I've tried to address in my career are the following. How does that actually happen? What are those mutations and why are they necessary for a virus to change its host? And if we understand that basis, is there anything that we can build on that knowledge that helps us prevent it happening? But actually, having just lived through the biggest pandemic that any of us have experienced, I think what's been very interesting to me as a molecular virologist is that once the pandemic arises, of course we want antivirals and diagnostics and vaccines. The 100 Days mission sort of aspires to that. But in the event, in the 21st century, we actually relied on non-pharmaceutical interventions to control it as well. And we needed a whole different set of knowledge to input the policy that made that happen. And then finally, of course, the question I think on everybody's mind is what's going to happen now? What happens next? What happens when a virus crosses from animals and becomes a human virus? Then what? Where does it go? And how do we understand and perhaps predict that? So for most of my talk, I will use influenza as the paradigm for a pandemic virus. Influenza is really a bird virus. Its natural reservoir are the wild waterfowl that exist in huge numbers and share their virus on the lakes where they congregate. It would be unlikely, I think, that a human would acquire infection or that a pandemic would be sparked by the crossing of a virus from a wild bird into a human. We don't really have much to do with those wild birds. But influenza can infect other hosts, including domesticated animals that we have a much larger interface with. And it's probably those animals, like the chickens and the pigs in these pictures, which pass the virus from the wild birds onto us. We also know that influenza has a track record of causing pandemics, so it's a good one to study because we can already see worked examples. There have been 4 influenza pandemics in the last 100 years. But as I've said, not all avian influenza viruses infect people and cause pandemics. That's because there's this big host range barrier. Remember your undergraduate lectures, viruses are obligate intracellular parasites. They utterly rely on hundreds of factors inside the host's cells that they infect. And those host factors will not be identical in different species. And in humans, they are different than they are in pigs, and in mammals, they are different than they are in birds. So we don't see frequent infections of people when they're exposed to wild birds, even though the ducks on that lake may be carrying flu. Sometimes we see those zoonoses, but again, as I'll explain, that doesn't always lead to a pandemic. Those can be dead ends. They can be very severe in the people who get infected by bird flu, but they don't always lead to pandemics. And it's really understanding the transition from stage 2 to stage 3 that I'm going to talk about most, because that's when we get a pandemic. And then after that, at least with flu, what we see is that the virus doesn't go away, and we transition to endemicity, and then we have to learn to live with the virus. The first host range barrier I'm going to talk about is at the level of the polymerase. Influenza virus is depicted on the bottom left here. You can see it's a very simple virus. It has a short genome of just 13.5 kilobases, split into these 8 separate pieces, which we call the segments. And each segment is an infectious unit. It's encapsulated up with its own RNA-dependent RNA polymerase, which is made up of the 3 proteins, PB1, PB2, and PA. They're what replicate the genome and make further viruses inside the cell. And all of that RNA is encapsulated by a nuclear protein. So that viral ribonucleoprotein complex exists 8 times over, if you like, inside the virus. But if that virus is an avian virus and it introduces that infectious unit into a human cell, it really doesn't work very well. There is incompatibility inside the human cell with the host factors that must also support the replication of the virus. And that was very vividly shown way back in 1977, actually in a single author Nature paper by my mentor, Jeff Almond. He was working on avian influenza at the time, and he showed that avian influenza viruses didn't replicate inside mammalian cells, but that you could adapt them in the laboratory, and you could then map where those mutations that conferred the adaptation were. And you can see on the left-hand side the plaques that are now forming by the mutated and adapted virus. And because influenza has that segmented genome, he could make what we call reassortant viruses, which switched segments from the one that didn't grow in mammalian cells to the one that did. And he said that there was just one gene segment, the PB2 segment, which was important for this. Now, I went off to New York after being in Jeff's lab, and I went to Peter Pelaez's lab, where they just learned to do what we call reverse genetics with influenza virus. That's the capability of making precise mutations in a recombinant virus. And one of the first things that we were able to do in my lab was to make a series of viruses that recapitulated that work from Jeff from all those years ago and mapped much more finely the exact mutations in that polymerase which were required to adapt the virus to human cells. And they mapped to the carboxy terminus of the PB2 protein. Now, that all made a lot of sense because we had seen that individuals who became infected with bird flu in the market very often, within 2 or 3 days, were selected for a mutation in the PB2 gene of a switch from a glutamic acid at residue number 627 to a lysine. That's a charge switch for those of you that know your amino acids well. And in fact, that same switch, we think, happened in the birth of the 1918 influenza pandemic and has been maintained in human influenza viruses throughout the 20th century. So this part of the PB2 protein was already known to be very important for host range determination, but nobody knew what the other side of the equation was. Well, where was the host factor? What was the host factor with which this polymerase needed to interact that was so important for the function of it? And I persuaded Jason Long, a postdoctoral fellow in the time, to pick up a project which we'd tried many years ago with Angie Lackenby and John Macaulay. And we picked it up again. What Jason did is we had a bunch of hybrid lines. These are hamster cells, but they've each got little pieces of chicken chromosome in them as well. So each of the different clones express a different array of chicken genes. And Jason screened them all one by one, looking for any which would allow the avian influenza virus to grow, because we reasoned that that clone had the piece of chicken chromosome in it, which was our missing factor. And I will never forget the midnight email I got from Jason on January the 15th, 2015, which just read, "We found it, we found it, we found it, we found it." It was the most exciting thing certainly of my career. And what he had found was a protein called ANP32, which stands for acidic nuclear phosphoprotein of 32 kilodaltons. And that made a lot of sense because influenza virus is an unusual RNA virus in that it replicates inside the nucleus. And what Jason was able to show was that putting that single protein into human cells enabled the avian influenza polymerase to work. So for the story to make sense, that gene needed to be different between mammals and birds. And indeed it was. When Jason scanned the databases, what he saw was that all of the flighted birds that he analysed had a gene duplication event, which meant that there was an additional exon and an extra 33 amino acids in the middle of that messenger RNA. And the spliced variant, or the longer variant, had this ability to support avian influenza, but the shorter variant found in mammals did not. That was all true except for ostrich. And I'm sure you're all sitting there thinking I wasn't expecting to have a lecture about ostriches. Ostriches definitely are birds, but they are more ancient birds than the flighted birds, and so they didn't get the gene duplication. And in fact, what had been reported is that when ostriches get infected by bird flu in ostrich farms, as sometimes happens, this famous mutation of glutamic acid to lysine at residue 627 in PB2 happens. So in other words, an ostrich looks more like a mammal, if you're an influenza virus, than it does a bird. So that all made sense, and we were very happy that we had found this factor that we'd been looking for a long time. Several years later, beautiful work from Erwin Fodor and Jonathan Grimes in Oxford have solved the structure now of this host factor in conjunction with 2 units of influenza polymerase. You can see one unit in blue on the left, that's the unit that's doing the replicating of the RNA, and then the unit on the right is the green unit that's going to be placed by ANP onto the nascent RNA. And then the ANP protein will also pick up the nuclear protein and pop that onto the RNA as well. And in this beautiful way, the virus is coordinating the assembly of its infectious unit, its ribonuclear protein complex. And what's now evident from the structure is the understanding of why that extra 33 amino acids, or the PB2 switch, is so important. So if you look at the structure there, you can see 2 red parts, I hope, down the bottom. Those are residues 627 in the PB2 of each of the subunit or each of the complexes. And you can see them here. This is the ANP32 in orange, and the tail of ANP32, which is acidic, dangles between these 2 red residues. Obviously, if this is acidic and your PB2 residues are glutamic acids, there's a charge clash there, and this complex is very unhappy. In birds, the acidic region is displaced by the 33 amino acids, which is inserted in the middle of the protein. In humans, when the virus adapts to humans, the charge switch alone of glutamic acid to lysine undoes the steric clash or the charge clash and enables adaptation in one foul swoop. So we now understand a lot about that first host range barrier and why some avian influenza viruses can adapt and start replicating in humans. But I need you to understand that that on its own is necessary but not sufficient to cause a pandemic. And that became very obvious when back in the early 2000s we had a lot of scares from bird flu viruses like H5N1 and then H7N9, which crossed into hundreds of humans and caused very severe disease, but never sparked a pandemic. There was one more block which the virus had not yet achieved, and that is transmission. And what I'm going to spend the rest of the lecture really telling you about is how important it is to try and understand what it takes for a virus to be carried through the air in infectious form inside these little droplets of respiratory secretion from one person to the other, because that really is what pandemic is all about. So for influenza virus to become airborne transmissible, it's quite a big change, because in the bird hosts, it's spread through the fecal route, through the water. And in humans, as we've said, it's spread in these little droplets. And so it's the outside of the virus, the spike proteins, which are called hemagglutinin and neuraminidase, which really control the way that the virus interacts with its external factors. About this time, in the early 2000s, when everybody was very worried about whether or not there was going to be a bird flu pandemic by H5N1, we realized that we needed better model systems to really study the way that influenza virus interacted with, for example, the primary target cells, which are the apical cells which line our nose and throat, the airway epithelial cells. And we reached out to Ray Pickles, who was working at Chapel Hill in the University of North Carolina, because he'd got an ability to culture those cells in an air-liquid interface. And he could make cultures with mixtures of cell types, both ciliated cells with beating cilia, and then goblet cells, which secreted a layer of mucus across the apical surface. And this was a good model, because these are the cells which transmitting influenza viruses have to learn to infect. We infected them in the lab with a human virus, and we stained. The cilia are stained in red, and the human virus infection is stained in green. And you can see many of the cells became infected, both the ciliated and the non-ciliated cells. However, if we took a recombinant virus, which we'd purposely back-mutated to make it look just a little bit more avian virus-like, you see the cell infection was much more restricted. It was limited to the ciliated cells, and the virus didn't spread. So this difference in tropism was very interesting to us and indicated a problem for the avian virus in that environment. The other problem that viruses have if they're going to transmit is mucus. We don't all make quite this much, but these airway epithelial cells, and all of us, of course, are overlaid with a layer of mucus, which is an innate barrier that protects them from invading pathogens like viruses. Now, influenza has a means to counteract that mucus. It encodes the neuraminidase protein as one of the proteins on its surface. Neuraminidase can chew up the sialic acid receptor decoys which are present in the mucus and enable the virus to penetrate to the surface of the cells below. But what we could see is that when we infected the cultures with an avianized virus, with an avian HA and an avian NA, it didn't spread, particularly at the lower temperatures of 32 degrees, which recapitulate the temperature of the human nose, in contrast to the human virus, which spread very readily. So in other words, it looked as if this avian virus NA couldn't deal with the human mucus. At that point, we realized that we needed to now move from working in those primary cell cultures to a whole animal model to watch viruses move from one host to the next through the air. And the model of choice was obviously the ferret. Ferrets had been long established as a good model for influenza viruses, and we were able to do some glycomic studies with Stuart Haslam at Imperial College to confirm that the types of sialic acid, which are the sugars, which are the receptors for influenza virus, that are expressed on ferret airway cells are more or less the same as are expressed on human noses and throats. So knowing that, we felt comfortable that our studies to look at transmission of flu would be bona fide, if you like. I'm very grateful to Kim Roberts and Holly Shelton, who were two postdocs in the lab at that time, who really pioneered our work with the ferret model. And I would point out that, of course, the ferrets tick all of these boxes, but they're quite hard to work with, and I'll say that we did have a few bites along the way. The way that you do a ferret transmission experiment is that you take a donor ferret and infect it with a known amount of virus. You can then expose a sentinel animal either in the same cage to look for what we call direct contact, or in an adjacent cage where the animals can't touch but they share the same air. So this would be the respiratory droplet type of contact that we think is important in sparking pandemics. Now, going back to the neuraminidase we knew was important for spread through mucus. And Dina Blumenkrantz, who was a PhD student in the lab at the time, had observed that the neuraminases of avian influenza viruses, particularly those adapted to poultry, were different from the human-transmitting ones. So neuraminidase is a tetramer. It has an enzyme in each one of these heads, and then it protrudes from the surface of the virus on what we'll call a stalk. You can see it here. the stalk of poultry-adapted viruses is for some reason shorter than of human transmissible viruses, and all of those viruses that were milling around in 2005 were poultry-adapted. So Dina used recombinant virology again to generate a human transmissible virus with the short stalk neuraminidase taken from an H5N1 virus from 2005. What she found is that when she had this virus and she compared it to the human transmissible one, it was unable to deal with human mucus. So she developed a neutralization assay where she harvested human mucus from the surface of our primary airway cultures and mixed them with virus. And the virus became totally opsonized, if you like, by the mucus and couldn't infect other cells. If you warmed the virus up with the mucus to 32 degrees, at which point the enzyme would become active, then the human transmissible virus could chew through the mucus and become infectious. But the virus with the avian neuraminidase could not. What Dina showed is that simply by restoring the length but keeping everything else the same, she restored the ability of the virus to counteract the effect of mucus. And that restoration of mucus digestion also correlated with the ability of the virus to spread through the air between ferrets. So neuraminidase is incredibly important for access of the virus to its target tissues and then for onward spread. But the other spike protein of influenza is hemagglutinin. And it's very well known, I think, that hemagglutinin is also different in the way it interacts in birds versus mammals with its receptor. So I've mentioned that the receptor for all flu is sugar sialic acid, but in the duck gut, it is largely linked to other things it's on, like other glycoproteins and glycolipids, by the biochemical alpha-2,3 linkage. That's a long, thin-shaped receptor. And if you look at the structure of hemagglutinin, you can see that avian viruses have a nice receptor binding pocket, which is long and thin and can latch on very well to that receptor. But humans predominantly express a different kind of sialic acid on their noses and throats, alpha-2,6 linked sialic acid. And so in order to be able to infect human cells productively, the influenza virus has to mutate the shape of its receptor binding pocket, and it can accomplish that by these two rather well-described mutations. This is the virus I showed you before, which we'd back mutated so that it could only bind to the alpha-2,3 linked sialic acids, and you can see how that sort of restricts its spread and tropism in the human airway cell. So what Kim did was take the ferret model again and take that recombinant virus and show that it did not transmit, even between animals actually in the same cage. But she wanted to really understand why. Why is it? Could we put some numbers on this and sort of get to grips with what was going on? So she then took further animals and infected donor animals with decreasing doses of virus to try to establish what was the minimal dose of virus needed for each different type of virus, the human one or the avian-like one, to actually initiate infection in the first place. Because she could see that both of these original animals were well infected, it's just that you didn't get transmission. So what she found was that, in fact, it took 40-fold more virus to initiate infection if you had the wrong kind of hemagglutinin. So if you think about what's going on when an individual is infected, they've got virus in their nose and throat, but that's not enough. You've got to get the virus out into these droplets, and then there's a distance over which the droplets are going to be carried in airborne particles. All the while, the infectivity of the virus is susceptible to all kinds of things in those droplets, as we'll see later. So the amount of virus that actually reaches the sentinel is much smaller. And if you have an avian-like virus which can't infect the sentinel at that kind of dose, then you won't get any transmission. So how much virus does it actually take during a transmission event to get infection? So we turned to reverse genetics again. We made two viruses, human-like viruses, which we knew would transmit, but we put a silent tag in one of them so that we could see it by sequencing. And then we infected the ferrets with a mixture of those two viruses at 9:1, and we asked, in the sentinel animals, do we see the mixture passing across, or do we see what we'll call purifying selection, indicative of a tight transmission bottleneck? And the answer was the latter, and we were able to estimate that around about 3 to 5 particles initiate infection during transmission of human transmissible viruses. And in fact, that's very much in line with what you see in the real world in humans, where people have done deep sequencing in household transmission chains. So we understood a lot more about how transmission actually works and how numerically you have to sort of get to grips with it. But the big question at the time really was back to the H5 viruses. Were the H5N1 viruses going to cause a pandemic or not? Could they acquire those important mutations which are necessary to carry the virus through the air from one person to the other and initiate infection at a low infectious dose? And a lot of people were looking at that at the time. There were some controversial experiments which uncovered that, in fact, it is possible that H5 can acquire those mutations, but in fact there was even more to it than we already knew. So for a start, when you engineer the two mutations to switch receptor specificity into the H5 hemagglutinin, those are very, very deleterious for the stability of the protein. So the H5 is not comfortable accommodating that change. Not only that, but in fact the actual codon usage in H5 viruses around that site mean that you need two simultaneous nucleotides even in one codon to make those switches. So you're already up to 3 nucleotide changes all at once. But finally, what was apparent was that even those viruses were not transmissible, and there were further mutations that were required to restore the stability of this HA protein. HA stability is really important for transmission. We were able to do some experiments that demonstrated that. We took our human transmissible virus and we engineered in, let's call it a loss of function mutation, to destabilize its HA and make it rather pH sensitive. We then infected ferrets with either virus. Both groups of ferrets became infected robustly and shed virus in their nose, but we then asked the ferrets to kindly breathe into this tube, which they did. And the virus in their airborne droplets passed through the tube and over cell culture plates with the lids off. And so infectious virus deposited onto the plates, and we could count the plaques and see how much infectious virus there was in the breath of the ferret. And for the human transmissible virus, we counted plenty of plaques, even out to the meter distance. But for the destabilized virus, there were very, very few plaques. And those that we did count, when we sequenced them, had reverted and become stable again. And this tells us that stability of HA is really important, and what we think happens is that because your respiratory secretions are already mildly acidic, as they pass through the air and water evaporates and they become smaller, the ions and the hydrogen ions in them become more and more concentrated and the pH falls. And if you have a virus which is rather pH stable, then it becomes inactivated in the air. So even if a person were infected and breathing out lots of virus, it wouldn't survive the journey to the next individual. So summing all of that up, I think we really know quite a lot now about influenza. We really do understand how avian influenza viruses climb this ladder from being locked in the avian host, perhaps through intermediate hosts, and then finally become these explosive pandemics. Knowing all of that detail, is there anything that we could or should do to try to stop this happening in the future? Well, as I've said, it's unlikely that an avian virus can do this all itself and get from here to here without a little bit of, let's say, help from intermediate hosts. And the type of intermediate hosts we're thinking about are really farmed animals. Now, there are two types of farming that goes on. This type of farming is farming animals for fur. And at the moment, the world is in the grip of a panzootic H5N1 again. And we had reports earlier this year that H5N1 virus had found its way into a Spanish mink farm. Now, it won't escape your notice that mink are very similar to ferrets, and mink, therefore, are quite likely to put some form of selective pressure on viruses to acquire the types of adaptations that could mean that an H5 virus prefers to bind to sialic acid receptors of the alpha-2,6 and become more transmissible. We know that mink farming does have some level of biosecurity issue because we saw in the COVID pandemic back in Christmas 2020 that mink in Denmark became infected with coronavirus, with SARS-CoV-2, that they spread it amongst themselves, that it adapted in them and passed back into people in the area. Thankfully, that outbreak was put to an end by the culling of 16 million mink, but there are many places around the world which continue farming these kinds of animals in these kinds of conditions, and I think we need to really address the biosecurity around there. And finally, of course, the hot topic of the moment is COVID and where did it come from? Raccoon dogs are implicated by the latest studies published in Nature last week from the Wuhan market, and raccoon dogs are another animal which we farm for fur. Other animals we farm for meat, and poultry, in fact, are the main source of protein for the vast majority of the world. But poultry are a potential intermediate host for future flu pandemics. Is there anything we can do using our knowledge that might stop poultry from passing flu to us? Well, using our discovery that the ANP32A protein of birds is key for replication of flu in birds, we worked together with Mike McGrew and Helen Sang at the Roslin Institute to generate gene-edited chickens. So this is early work from Helen, and you can very clearly see which one is gene transgenic. These are our chickens, and I don't think anybody in the audience could tell me which one is altered in chicken ANP32A and which one is not. It's perhaps pertinent that we start thinking about whether we do this and how we do it, because this year the UK Parliament passed the Genetic Technology Precision Engineering Bill, which means that it is now legal for people to pursue these kinds of approaches, if you like, in the UK for both plants and animals. So we looked at that structure that Owen Fodor made, and we looked at ANP32 proteins across species and asked, were there very simple edits that we could make? We didn't want to take away the whole gene from the host because obviously all of these host factors don't exist in these animals for the benefit of the virus. They're there doing very important functions, and so farmers won't want to farm chickens if they're not healthy and doing what they should do. So we made a minimal edit in the ANP32A protein of just 2 amino acids, which we knew would stop the influenza virus polymerase being able to recruit it for its encapsulation. And then we took those gene-edited birds down to Pirbright and challenged them. Holly Shelton was working at Pirbright at the time, and she challenged the birds with a low pathogenic avian influenza virus. 10 to the 3 infectious units. The wild-type birds all got infected and transmitted their virus onto most of the birds that were incubated in the same incubator with them. The gene-edited birds, one bird got a little bit of infection and didn't transmit at all. So we then decided that we really needed to test this with a very robust challenge, and Holly inoculated 1 million infectious viruses into the sets of birds. This time around, all of the wild birds got infected and they all transmitted, and none of the sentinel birds we put in the same incubator that were gene-edited acquired the infection. However, direct inoculation of 1 million infectious viruses into the gene-edited birds did result in 5 low titer infections. No transmissions, but one of those low titer infections did transmit onwards. So I'm pointing all of this out in complete openness to say, look, I think we've got more work to do. This is not good enough. We need resistant, not really resilient birds. And that's going to be important in the future. How many, if we go forward with this approach, how many layers of gene edits would one require in order to make this more robust than it currently is? But equally, from a scientific point of view, it's very interesting to see what's going on, because when we sequence the viruses in these breakthrough cases, we can see mutations in the influenza polymerase. But what's really fascinating is that they largely don't map to the interface between the ANP32 protein and the influenza polymerase, but they actually map to the interface of a different dimer, a symmetric dimer, also described by Owen Fodor's lab, which we know is very important for the replication of the genome in some of the other stages of replication. So I think we're going to learn an awful lot about how influenza virus works by looking at how it can work in the presence of slightly altered host factors. And meanwhile, we were busy doing all of that, and at the end of 2019, along came this virus, completely unexpected. I'd been sitting on committees like NervTag. We were well prepared, I think, for an influenza pandemic. We had antivirals, we had vaccines, we had some validated diagnostics, and then a coronavirus emerged. We had seen a coronavirus, of course, in the early 2000s, SARS-CoV or SARS-CoV-1, but already it was obvious in the early months of 2020 that SARS-CoV-2 was a different beast. It was transmitting much more efficiently than SARS-CoV-1. Thankfully, although WHO were informed rather late about SARS-CoV-1, it was contained because people were only infectious when they were overtly sick, and therefore it was possible to identify people who were infected and put them into isolation. In contrast, although WHO were informed earlier and the world responded much more rapidly, it was all out of the bag. By the time we got to 200 days in, we were into millions of cases, and I think it was, at that point, uncontrollable. This wasn't because SARS-CoV-2 was using a different receptor. It uses the same receptor as SARS-CoV-1, which is well known now to be the human ACE2 receptor. And indeed, Neeltje van Dormalen, working at the Rocky Mountain Labs, showed that both viruses were equally stable in the air as well. So it wasn't that it was more stable in the environment, but it really was that it was transmitting better. And what that means is that some people can be infectious to others earlier in their infection, perhaps before they're even overtly ill. And it's very difficult to then find them and control the outbreak. Why is it? Why is SARS-CoV-2 more transmissible than SARS-CoV-1 was? Well, if you look at the sequence, of course, there are many changes, but one of the most obvious differences between the two was this famous insertion in the spike protein, the furin cleavage site. So coronavirus spike proteins are made as one long protein precursor, and then they're cleaved at the S1, S2 junction. That primes for further cleavage at what's called S2 prime, and that S2 prime then enables the virus to fuse its membrane with the host cell membrane and enter the cell. And the presence of a furin cleavage site in SARS-CoV-2 means that the virus particle is already cleaved at that position as it exits the cell it's just been in. And therefore it arrives at the cell surface of the cell it's about to infect, primed and ready to go. We then think that a cell surface protein, TMPRSS2, TMPRSS2, can readily snip the S2 prime site, and then you initiate infection very rapidly. So how did SARS-CoV-2 become a pandemic? This cleavage site leads to this enhanced replication in airway cells and efficient transmission. And these 12 letters encoding those 4 amino acids of the cleavage site really did, I think, change the world. Rapid entry of viruses into cells, even potentially avoiding endosomes, can be very advantageous in the right circumstances. And this, I think, is what SARS-CoV-2 was doing. Viruses which don't get primed by that furin cleavage site have to wait and enter endosomes to be activated at the S1, S2 junction before they can then fuse. And the problem for viruses entering cells through endosomes is that we have evolved an innate immune system that has several restriction factors that sit in endosomes watching out for viruses that like to invade us in that way, including things like the IFID-M proteins. And so by enabling that rapid cell surface entry, SARS-CoV-2 had avoided all of those restrictions. We were able to go back to our favorite model, the primary human airway cultures, and using a mutant which had arisen in the lab, which lacked the furin cleavage site, shown in blue, we could show that it was rapidly outcompeted in those primary human airway cells by the wild-type SARS-CoV-2, shown in orange. We also, of course, had some experience at doing transmission studies, and we had some ferrets who had been lined up for a flu transmission in early 2020, and we had all the kits ready for Ji Zhao or Jay, our postdoc, to infect those ferrets and look at transmission. Now, the ferret is not a good model for SARS-CoV-2, so we didn't get 100% transmission with the wild-type virus, but we didn't get any transmission at all from the deletion mutant, really suggesting that furin cleavage site was key for the emergence of the pandemic. So we know, really, why SARS-CoV-2 managed to become a pandemic virus, but as I said in the introduction to this talk, at the end of the day, all of that molecular knowledge that we have really didn't make much difference to the way that we tried to control the pandemic, and we resorted to largely non-pharmaceutical interventions, asking people to stay at home, test and trace, and then in the latter parts, sort of more diagnostic tests. I think what we have to reflect on now is whether or not people would do that again, and how good was our scientific basis on implementing that policy. One of the things I think we really wanted to know much earlier than we did was the detailed kinetics of infection, who was infectious to others and when. And we've been involved in a number of studies monitoring virus in the environment and also monitoring virus in households, as I'll describe. This is a section from a household study that was led by Ajit Lalvani. Each one of these boxes is one of 57 people who were traced in households as they passed the COVID virus from one to the other. There's a number of different variants listed here. and we've measured how much virus is being shed in their nose and throat in terms of viral load, RNA in orange, and infectious virus in purple. And you can see that in general, one can put this all together and come to conclusions which would better inform policymakers. For example, most people shed infectious virus for just 5 days, and that's infectious virus actually in their nose and throat. It doesn't actually mean it's coming out into the environment. So we probably could have allowed people, let's say, back to work a little bit earlier. Also, there's some very interesting observations. 2 of these individuals are still shedding infectious virus even on the last day of swabbing, and those 2 individuals happen to be 2 people with type 2 diabetes. Now, there's a lot going on in the literature about long-term infections, chronic infections in immunosuppressed people and how that may lead to future variants of concern. But I don't think that we always consider that people with type 2 diabetes are also at risk as much as we do from these viruses, and that they can be shedding virus for a very long time and perhaps driving some level of ongoing evolution. We were also involved in a human challenge study with a first wave SARS-CoV-2. 36 volunteers came and had 10 infectious units instilled directly into their noses. Of those, 18 became infected and had very mild symptoms, but we were able to do very detailed studies about the virology in them and also what they put out into the air, in their breath, on their hands, and on swabs. And what we could largely see, for example, is that if everybody took a lateral flow test every day and then stayed at home only when they were positive, we would have stopped 90% of the virus actually being emitted into the environment that could potentially be passed on to another person. And I really think one of the things that the COVID pandemic has shown us is the power of home-based diagnostics and giving people the information themselves. Who would have thought that so many people could actually do a nose swab and perform a lateral flow test, as has happened? And this type of approach also enabled huge epidemiological studies, such as the REACT study that we're also part of. And I do put forward here that improved diagnostics to detect who's infected early could also help us use antivirals much more effectively in the next pandemic, because the problem with using antivirals against acute respiratory infections like flu and COVID is that you always get them too late to really control viral load in people. But if people knew the day that they were first positive, then this could be a really good way of treating people and also preventing transmission. The other thing that we saw in our human challenge study is that just 2 of the 18 infected people emitted 90% of the virus that we detected in the environment on just 2 days. This is real evidence, I think, for super spreaders and super spreading events, but we don't know how to identify those people, because we could then focus our efforts on containing transmission much more accurately if we really knew who they were and who they were going to be. And then, of course, the pandemic went on, and the transition now from pandemic to endemic, variants arose. I think many of us realized that there was a huge amount of fantastic sequencing coming out from COG UK, led by Sharon Peacock, huge efforts to understand how the virus was evolving. And I'm very happy that a group of virologists in the UK came together to work as a consortium to ask how to interpret all of that sequence analysis and understand what the sequence changes were meaning for the phenotype of the virus, such as immune escape, both against acquired and innate immunity, but also transmission, severity, and whether or not the virus has the potential for infecting other animals. In our lab, we've had to learn to use a different animal model. They're smaller and cuter, but they still bite. And we think that the Syrian golden hamster is a good model for human disease. This is very beautiful work from Tommy Nyberg on humans at the point at which Omicron was just beginning to displace Delta from the community. And what this is showing is a hazard ratio in various different age groups, from the very young to the very old, as to how likely it is that a person will end up going to hospital if they get infected with Omicron versus Delta. You can see it's much, much less likely for nearly everyone to go to hospital if they have Omicron, suggesting that Omicron itself is inherently milder. I'm not saying mild, I don't think it's a mild disease, but it is milder in humans than Delta was. And you can see that playing out in the weight loss curves in the hamsters. As you infect hamsters with just 100 plaque-forming units of wild type or alpha or Delta, they all lose weight, but the Omicron variants don't cause weight loss in hamsters. So what we really want to understand now is whether or not this sort of public myth, if you like, that as a virus adapts to human hosts, it will become milder as it becomes more transmissible. Is that going to hold for these viruses? And I think in order to really understand that, we have to understand the exact molecular basis of the various phenotypes that Omicron shows. And I think some of you in the audience may have already seen this work before today, but we're very pleased to collaborate with our colleagues at the CVR, the Centre for Virus Research in Glasgow, who are very capable of making recombinant SARS-CoV-2 viruses. And Jonathan took Omicron and Delta and put them into our two models. We use primary human nasal epithelial cultures still as our best model for what we think a human transmissible virus should grow in. And you can see in the solid purple lines here that Omicron grows very rapidly in those nasal cultures, and Delta more slowly. In Calu-3 cells, which are an immortalized cell, but we think they approximate more to the lower lung, the Delta virus is much more replicating quickly, and Omicron is in fact attenuated. And so this trend for efficient replication in the nose and less efficient replication in the lung tends to sort of support the concept that as you get more transmissible as a virus, you're less likely to cause at least a viral pneumonia. And you can see with these recombinant viruses that largely speaking, that phenotype is mapping to the spike protein. What we now need to really understand is which pieces or which changes in spike of Omicron are really determining all of that. And meanwhile, as I've mentioned, out there in the world, there is clade 2344b, H5N1, causing this unprecedented panzootic. You will have seen in the news tragic pictures of wild birds dying. We're losing species that are rare, seals and sea lions and other aquatic mammals being infected, foxes and otters here in the UK, and even dogs now in Canada. And again, I think the way to tackle these problems that have some emergency need about them is to come together, such as the FluMAP Consortium is doing at the moment. So personally, I'd like to make 3 reflections really on doing science in pandemics or for pandemics. The first is that I think understanding transmission is really key. Pandemics are just about a virus that can transmit really, really well. And we don't really understand enough about that. And that can come both from doing more virology, but also more interaction across different disciplines, physicists and aerosol biologists that understand how these clouds work. Secondly, I think that certainly members of advisory boards and people who are thinking about future policy must think outside of the box. We hadn't really thought carefully enough about coronaviruses causing pandemics, and I think there are other viruses out there which are relatively understudied, including whole tranches of paramyxoviruses, which we know can become airborne transmissible between humans, but we rather dismiss. So I think we need to invest more in understanding how they work. And also, I don't think before 2019 anybody would have thought that, as you say, the public were as familiar with lateral flow tests as they are now, and perhaps we could think about how to incorporate that learning, that communal learning, better for next time around. And then the final thing to say is really, I think that in virology, we're in a very dangerous place at the moment. There is a lot of public mistrust of science. Many people think that COVID was created in a lab. Other people think that virologists, including myself, should not be allowed to do what we do because they don't understand why we do it and how we do it and what the regulations are that are in place. And we need to make more effort both to communicate that to them and to ask ourselves and challenge ourselves about all of those things. So I'm going to finish there. Thank you very much for listening to me. I just want to say the biggest thank you, actually, to my wonderful husband, Dan, who's been with me every step of the way. Thank you.